Morning, everybody. It looks like we still got a few folks that are going to be coming in here in the next few minutes or so. Hold on one second. Okay, so we are going to go ahead and get started. So, plan for today, I'm going to go over our first assignment a little bit, not like too much, just because we do have a lot to get through lecture-wise. I'm going to go through just like a couple of points that I saw that we'll want to improve for next time, which is going to be this Sunday. And then I will detail a little bit about your next assignment, which, like I said, will be due Sunday night at midnight, like they pretty much always will be. What was I going to say? I thought you'll do a good job on the first one. The second one, pretty similar, but we're just going to be looking at some stuff that's more specific to chapter 2, which is what we're going to start today. And we're going to do this just like we did last week, probably do half an hour, 35 minutes ish, take a break, and then we will come back on the same Zoom link after our break, which will last 5-6 minutes, something like that, just for enough time for everybody to go pee and collect themselves, whatever you got to do. Okay, make sure I got all my stuff pulled up. Okay, so going back to assignment 1 for a second, which is the one that we just turned in. The biggest thing, and I didn't specifically request you guys do this, and that was kind of on purpose. I always want, when you include something in an assignment like this, so like if I do, sorry, absentmindedly moving the screen. If I talk about behaviorism and I say, okay, well, and I'm talking about ADHD or something like that, or, in this case, I was talking about in-person remote learning. And if I say something like, okay, well, it's understood that ADHD is characterized by inability to pay attention, or that remote learning, there's a tendency for kids to spend more time goofing off, doing things like that. So those are assertions that are supported by research. But if I don't include the research, if I don't include the study where I got that information, I'm just kind of, spouting off information without any support, and that's not good. So what we want to work towards is eventually, and I'll probably, when we get to assignment 2, which I'll explain in a second, we are going to be looking toward, if you talk about an idea, if you mention a theory, if you, say something like, if we're talking about how neurotransmitters affect generalized anxiety disorder, you need to say, okay, well, this study, this, the NIH says this, whatever, the APA says this about generalized anxiety, just so, it's supported by research that people have already done, because there's a good chance that somebody has already looked into that and you can take advantage of that. And that is going, like I said, we're doing all these assignments to sort of build up to the paper at the end of the semester. And So sort of building from point A, which was coming up with an idea. The next piece is going to be zooming in a little bit more on one particular issue. So from the following disorders, you are going to pick one of them, and then you are going to answer 3 questions about whichever disorder you choose. Okay. So we're going to talk a lot today about like neurotransmitters and neuroscience and things like that, and pretty heavily involved in all of this. When you're choosing between ADHD, stroke, aphasia, GAD, GAD, depression, and then seizure disorders like epilepsy. So once you've chosen that, you are going to come up with the answers to 3 questions. The first one is, what is it? So what sort of symptoms typically characterize that disorder? What are its identifying features, which can kind of be the same thing? How is it understood in popular media? And does it have any comorbidities? So we're going to talk a lot more about what this word means. But basically what it means is it frequently diagnosed alongside other disorders and conditions? So for instance, if you have autism spectrum disorder diagnosis, people with ASD typically also have a comorbidity like ADHD. It's not always, but very, very frequently. So if you have ADHD, there's a good chance you'll have anxiety disorders or depression, things like that. All right, so the second question is, how does it work? So which neurotransmitters does it affect? We'll talk about what a neurotransmitter is today. How does it interrupt your brain chemistry? And then the third one is, how is it usually treated? If there is a prescription treatment for it, so if it's being treated through psychopharmacology, so being treated through some type of pharmacological intervention, like a pill, what does that drug do? How does it affect all this stuff up here? Are there any alternative treatments? Are any of them effective? So that is basically how this is going to be set up. And then I give you a sample prompt. And like it says here, I said, include references to peer-reviewed research with citations, be about a page long, and should be formatted like this. Okay, so when I say citations, this is all I'm asking for. So basically just give me the name of the author and the year it came in. We're not going to do a formal works cited for this because that is, this is a page long paper and that would just take too much time. We will do a formal works cited for the actual paper that we do at the end of the semester. And we'll practice doing that before we get to that point. So yeah, okay, so we just had a couple people come in late, but if all of these, I should mention this now, all of these lectures are up on our class YouTube page. I will put the link to that in our, where my information is on the front page of the class, in case you don't have it already. But every lecture we've done is already up there. Okay. So that is assignment 1 and 2. If you have any questions about those, feel free to email me about them. Your test is still kind of up in the air about what I want to do about that, because we kind of got a late start to the class. We will have all of the information ready, because next week we'll finish chapter 2 and do a Kahoot. So I got to decide where I want to put that test, but I will have that to you. Because originally it was scheduled for next weekend, the 13th, but we may not be ready for that. I'm not 100% sure. So I will, when I make a decision about that, it'll be the next few days, and I will let y'all know. Okay, but I will say that all of our tests are open note, and so don't freak out. And everything that we talk about, everything that is on the test will be stuff that we have talked about in lecture. So it won't be as horrifying as it sounds. Okay. So with that in mind, let's talk about chapter 2. Woohoo! All right. Please launch. All right. So chapter 1 was pretty history heavy. So we talked a lot about old dead white guys and the ideas that they had. Chapter 2 is not going to be like that nearly as much. I think we have one dead white guy who we talk about a little bit, but it's mostly, if you're a biology person, this is going to be kind of your jam, because we're going to be talking a lot about electrons and neuroanatomy, things like that. And if you're not a science person, you're more like me, and you like the history and that kind of stuff, just bear with us, because I promise it is interesting. It's just very different from what we've talked about so far. All right, so tentative plan. overview for this is we're going to introduce the brain, talk about neurons a whole lot, talk about other structures that help the brain, kind of do all this stuff psychologically, to talk more about the brain. It can be a lot of brain stuff, and then digging a little deeper on all of that stuff. Okay, so okay, well, sip water here. Your book, as I mentioned with chapter 1, loves a case study. So in this case, we're talking about this gentleman, Brandon Burns. He was shot in the head on a tour in Iraq, which you would think would be fatal, but it actually wasn't in this case. It's just what we would call a traumatic brain injury, or TBI. So the treatment in this case included surgical removal of part of the skull and a third of his brain. So again, conventionally, we probably think that taking a third of your brain out would be pretty fatal, and it might have been. But in this case, this gentleman survived, and through a lot of therapy actually came back and lived a pretty normal life. But it was obviously a little different, because the question here, how would this type of injury impact your life? Quite a bit, and we'll explain why over the course of this. Okay, so starting with the nervous system. It's a term you've probably heard before. It's a communication network that conveys messages throughout your body using electrical and chemical processes. Okay, so electrical, we're going to be talking about nerves and neurotransmitters. Chemical processes branches out a little bit into like hormones in your endocrine system. We'll get to that later on. Your brain, you guys know what that is. Consists of a whole bunch of nerve cells. And when we talk about like, me, moving my hand, me speaking, all of these things are happening like hundreds of thousands of times, like per second. All these things is just in massive quantities to be able to just like move and do normal human stuff. Provides the power to think in complex ways and differentiates us from most other organisms. So we have very well developed brains in a way that most other organisms don't. And we'll talk about why that is and how that actually works when we get to the actual brain anatomy later on. So neuroscience is the study of the brain and the nervous system, integrates information from a whole bunch of different fields, because it turns out psychologists are not the only ones that are interested in this. So if we're interested in primarily how neuroscience affects behavior, somebody who is a doctor or a nurse is treating it from a sort of a medical lens is also interested in neuroscience, because that can affect a bunch of different other things about the body. But biological psychology, so we've talked about our biological perspective in chapter 1. So that focuses on the brain and how other biological systems influence human behavior. So there's your connection between this idea of neuroscience, the exact mapping out of what is going on in the brain neurologically, to how we actually behave. This is a subfield of psychology that we've already discussed a little bit. We're going to talk about it a whole lot for chapter 2. Okay, so there were a lot of early attempts to try and understand some of this. We don't, back in the day, we're talking about, the 1800s, they didn't have the kind of radio imaging that we do now. This began with this guy. This is probably the one old white guy that I talked about that you will now have to know. Franz Joseph Gall came up with this idea of phrenology. So this is basically the idea that sort of like the different contours of your skull and the way that your skull was structured would determine things about your personality and how intelligent you were and what sort of work you were cut out for. And that was, of course, nonsense, and that science did not last. But it sort of started us off on the direction towards understanding that certain parts of the brain were responsible for certain functions. And again, I can't emphasize this enough that it was a load of garbage. Phrenology is not a real thing. Ablation is another of these attempts. You can see there's crossover in the timeline there with Franz Joseph Gall and Pierre Flourens. Ablation is destroying parts of a living animal's brain, determining whether some functioning was lost thereafter. So we've talked about experimentally how you can control for a variable and figure out, okay, well, is this actually affecting this thing? Ablation is sort of a pretty brutal and inhumane practical application of that idea. So the basic idea there is that they would take out parts of like a cat's brain and figure out what kind of behavior was affected from removing that bit. It would also kind of serve as a therapy. So if they, if someone is experiencing seizures or significant mood swings or something like that, they would take out specific parts of their brain and see if it did anything. And I should emphasize that these creatures, animals that were going through these experiments were still alive. And so that obviously is not something that we still do today because it's not very humane. Today, we have a lot more imaging and recording technology that helps us actually look at what's going on very specifically. One of these ideas, optogenetics, thing here down at the bottom, sort of the idea that you could turn off certain parts of your brain using light, individual brain cells, and then you could observe what happens when they manipulate the activity of those cells. So it's the same principle behind ablation, where you're sort of manipulating one area of the brain, switching it off and seeing what happens, but without all of the brutality, which is good. Okay, so some ways to scan the brain. There's a whole bunch of them. So if you've ever had a concussion or some like serious sinus infection or something like that, you may have gotten a CAT scan. So a CAT scan is computerized axial tomography. Okay, so you can see cross section of the brain. You see the ventricles here in the middle. They form the butterfly-shaped dark spaces. Those are the ventricles. You can also get an MRI if you've ever like torn your ACL or broken your ankle or something. You might have gotten an MRI to see what the damage was. MRI uses magnets. It's a magnetic resonance imaging. So same, like very similar, like looking scans, because you're also producing like a cross section from the top of your head, but with more detail. Those magnets are mapping out what's going on in the brain. So if you have a significant brain injury, you'll be able to see exactly which parts of the brain were being affected. All right, so you have an electroencephalogram. I was actually had a conversation with one of my students in my other classes. So this can also be used as a therapy. So if you've ever, like I think the example they used was their kid had ADHD and they were using this electroencephalogram to sort of map what kind of brain activity was going on while they were watching television, and they would use that as part of their therapy. But it's basically just mapping out radio waves, electric waves, to see which parts of the brain are more active in certain activities, based on where these cathodes are on the top of somebody's head. Then you also have a PET scan, which is probably the most often used one, because there's the positron emission tomography. That's what PET scan stands for. So this is, you get this radioactive tracer in your blood, and then based on where that radioactive tracer gets sent to the parts of your brain based on the activity you're doing, they can map out which parts of the brain are being utilized. Similar thing happened in fMRI, so it's just more advanced MRI technology, where you can see very clearly where certain activities are happening in the brain based on what's going on outside of it. All right, so this is where it starts to get really important. So if you had to memorize one thing over the course of this whole chapter, I would probably say that the neuron anatomy is right there at the top of the list. If you've ever taken a biology class, this is going to be review probably, but we are going to talk about it through a psychological lens because it's very important. All right, so what's a neuron? A neuron is a specialized cell that communicates with each other through electrical and chemical signals. So when we talked about the brain, you have a whole bunch of these, especially in the parts of your body that are more specialized. So we'll talk about the homunculus a little bit later, but so like your hands have a lot more advanced neural connections than your feet do, because you can't really like write with your feet or type on a keyboard with your feet. You don't have the kind of specialized fine motor skills in those areas that you do places like your hands or your mouth. Anyway, this neural cell. So if you can see on the, everything starts in the soma. The soma is here on the left, this big circle bit. It's got the sort of like jelly center looking thing. So the soma is the cell body. The soma is where all of the DNA of the cell is that determines what type of neuron this is, because there's a whole bunch of different types of neurons based on where they are in the body, what kind of function you need them to perform. But all that information is going to be in that cell body. It's all like the mitochondria, all that kind of stuff is right in there. So the dendrites, you can see these little like finger looking things that come out of that cell body. So the dendrites are there pretty much exclusively to try and pick up signals from other neighboring neurons. So that is their function. They will feel out there in the ether to see if they pick up a signal, and if they do, they will send that signal to the cell body. It'll pass down the axon. The axon is that sort of tail-looking thing with all of the little circle guys on top of it. And the axon has a couple of different important things on it. So the myelin sheath, so you can see this sort of like, kind of looks like a subway. Like the whole axon looks like a subway, and that's basically how it works too. But the myelin sheath is this fatty substance that hangs out on top of the axon to help facilitate the reaction. So it looks and functions like insulation around an electrical wire, just makes that reaction happen faster, more efficiently. The node of Ranvier is the little space in between the myelin sheath. We'll talk about what it does a little bit later. And then not pictured, the axon terminal is all the way, like if you kept looking down over in this area, there would be an axon terminal. The axon terminal, like the name would suggest, is the end of the axon. And then from there, once the signal reaches the axon terminal, the dendrites of another neuron would pick it up, and then it would it's like a big chain. And so if signal starts at your hand, gets to your brain, there's neurons in your hand, send information, just keeps passing that information along up to your head. And it happens very quickly, obviously, because if if your cat nibbles your finger, you feel that pretty instantly. But that's just a neural signal that happens, starts in your hand, goes to your brain, and then your brain determines what to do about it. Synapse, I should mention that. Synapse is where two neurons connect. There's a lot of different anatomy and important stuff that happens there as well. All right, glial cells. Glial cells are very, very important. We don't talk about them a ton. We could go into way more detail because there's a whole bunch of different types of these things. But what you guys need to know is that the glial cells support, nourish, and protect neurons. That's pretty much their sole job. And that myelin that we talked about, sort of the goo stuff that's on the top of the axon that makes it go faster, that comes from the glial cells. So the way I've described this in the past is that, if we got any basketball fans in here, if your neurons are sort of like your Steph Curry or Klay Thompson, the glial cells are like the Draymond Green, like the glue guy, the one that does all of the dirty work so that the superstars can shine. That's basically what the glial cells are responsible for. Glia means glue in Greek, and there's a lot more glial cells than there are neurons. And without glial cells, your entire nervous system would fall apart. If they deteriorate, you're in big trouble. Okay, this is where it gets a little technical. So bear with me. I would highly recommend going and investigating this on your own because there is a lot of stuff here. Okay. So communication within neurons, this is their entire function. Okay, neurons are chemical communicators. A neuron is surrounded by and filled with a fluid containing particles that have an electrical charge. Okay, so that electrical charge at its resting potential, so where nothing is happening, is -70 millivolts. So you need to remember that number. This membrane, so the outside of the neuron that either things will pass through or won't pass through, is selectively permeable. Okay, so you can either have an impermeable membrane, a permeable membrane, or a selectively permeable membrane. So a selectively permeable membrane means that some stuff can get in, but not everything. We'll talk about why that is and how that works in a second. But getting this to actually do something, because your neurons don't just like fire all the time, or you would just be like moving constantly and it would be exhausting. So you get stimulation from a neighboring cell, going back to our dendrites. So if the dendrites pick up a signal from a neighboring neuron, this process will start. Channels in the neuron's membrane start to open up, starting at the dendrites, which allows positive ions to enter the cell. Okay, so it starts at -70, but as those positive ions start to come into the cell, it raises that charge. And if it gets over -55, the cell fires what we call an action potential. So an action potential is just where a neural signal is actually passing. You can see in that chart, in between representing the membrane, It goes from that -70, and as soon as it gets to -55, it fires, goes all the way up to, in this case, +30, and then comes back down. That is something known as the all or none principle that we'll talk about a little bit later. So what happens after it actually fires? Because like I said, your neurons aren't just firing constantly, because then like you would just be moving all the time and it would be exhausting. This process is very expensive nutritionally and in terms of like using up energy. So your body doesn't want to, fire action potentials any more than it has to. So what happens after this sudden jump in voltage? It comes right back down. Okay, as soon as that action potential fires, it gets to that positive charge, it comes right back down to its resting potential. So there's this idea of spreading activation that as it goes down the axon, as it passes from neuron to neuron, that charge would continue if it was strong enough, if that signal is important enough for it to actually fire. And you can see it sort of looks like a radio wave, and that is functionally exactly how it works. There's different types of signals. Excitatory signals tell the neuron to fire an action potential, and inhibitory signals do the exact opposite. You have to have more excitatory signals than inhibitory signals for that action potential to occur. You have to have more positive than negative for that to actually go through, which makes sense. Okay. All right, so I mentioned the all or none principle. That is the idea that a neuron either fires or does not fire. There's no like halfway point. There's no like, okay, well, I'm only going to fire a little bit. It either goes all the way or it doesn't go at all. And key point here is that action potentials are always the same strength. Okay, so we're going back to our chart here. You can see that peak is always the same regardless of the signal, because the strength is always the same. So the question then becomes, how do we, because like I mentioned, like your cat nibbling your hand earlier, that's not that strong of a signal, but if you accidentally cut one of your fingers off, that's going to be a very strong signal. So how do we determine which one is which? How do we differentiate between those two? Being able to do that is very important. So if you don't, if you have some sort of neurological issue that would prevent you from distinguishing stimuli like that, that's going to be a problem. But how do we differentiate them? So neurons will fire more frequently with strong stimuli. So if you can imagine, think about it like an EKG, like you've ever seen like a heart monitor. So if it's just like a very slow, like normal looking signal, like it will just kind of, there will be a peak and then go back to the resting potential, peak, back to the resting potential, everything will look pretty normal. That's a pretty low strength signal. But if it is something like, you step on a Lego and it really hurts, that's going to look like this. Okay, it's going to be a whole bunch of the signals happening. And another key point here, more neurons are going to get that signal. So it's going to be more localized if it's a small signal, but it's going to be spread out throughout your body more if it is a strong signal. Hey, do you need something? Oh, okay. Just love. Okay, so your myelin sheath, mentioned that. That's what's pictured here. In this graphic, it's a fatty substance that envelops, it insulates the axon, facilitating faster reactions. That's exactly what it does. It just kind of covers up this whole reaction and makes sure that it is running smoothly. Hold on, there. All right, so we're getting to neurotransmitters. So actually, let's go ahead. This part is important, and we have just thrown a bunch at you. So we are going to go ahead and take our quick break. So it's 9:14 now. So we are going to come back at let's say 9:22, just to pick an ugly number because I'm the teacher and I can do that. All right, I'll see you guys in a few minutes.